Welcome everyone to today's program. I'm Heather Punke, Managing Editor of Becker's Infection Control and Clinical Quality. The program will begin with a presentation, and we will have a question and answer session following completion of the presentation. You can submit any questions you have throughout the webinar by typing them into your control panel in the space labeled Enter a Question for Staff and clicking Send. Our presenters will attempt to answer as many questions as they can during the time we have, and will follow up on questions they don't have the opportunity to address. You'll receive an email within about a week following the webinar that will include instructions for how you can download a copy of the presentation. You will also receive a follow-up email shortly after completion of the program. You can submit your feedback or any additional questions at that time, but that email will not include the presentation. It is now my pleasure to introduce today's presenters. Stephen M. Kovach is the Director of Education at Healthmark Industries located in Michigan. He has been in the hospital field for over 37 years. He started his career as a sterilization orderly in 1975. Stephen is active both on the state and national levels of various organizations, having held many positions. Stephen was the Educational Chair for AORN Specialty Assembly for SPMM between 2006 and 2010. He is a voting member on various AAMI committees and has been an instructor at the community college level and published many articles varying in subject matter from perfusion to the importance of cleaning surgical instruments. And Mary Ann Drosnock is Manager of Clinical Education for Gastroenterology at Healthmark Industries, where she is responsible for providing expertise and resources in regards to endoscope reprocessing. Mary Ann often presents relevant materials at national, regional, and local professional society meetings and corporate workshops on effective device reprocessing and infection prevention in endoscopy. Prior to her position at Healthmark, Mary Ann was manager of the Infection Control Program for Olympus America, Inc. for 5 years. Prior to Olympus, Mary Ann worked for 9 years in the pharmaceutical industry as a microbiologist and taught microbiology courses at the college level. As of 2014, she attained the status of Nationally Certified Registered Microbiologist through the American Society for Microbiology. It is now my pleasure to turn the floor over to Steve to begin today's presentation.
Next slide, please. Hi, this is Steve Kovach, and I want to welcome you today. I just want to take 2 slides to tell you a little bit about Healthmark and our company mission, and it's up there on the slide, and we try to find innovative and cost-effective products to our customers throughout the healthcare industry. If you go to the next slide, please. We have a policy that it's really focused on understanding our customers and delivering the highest quality products and support. And today is one of those. We have a commitment to professionalism and mutual respect, but also to educating our customers. And this webinar is one of the ways we're trying to educate our customers, and we're going to start talking about flexible scopes. Next slide, please. As with any program, we have objectives. And we have 4 we'd like to get to today. We want to review the latest information from various organizations on flexible endoscopes in the last year. We want to discuss the key provisions that are found in the new standard ST91 that Amy came out with this year. We also want to review ongoing and updating the document that's going to take place within ST79. And lastly, we want to work and try to share with you some best practices for processing flexible scopes. Next slide, please. So a lot's been going on if you see this slide here. I'm going to now try to set up the importance of why ST79 has been made. And if you look here, there was in Seattle, again, a couple years ago they had an issue. I'm sorry, ST91, I said 79. I'm so used to ST79, excuse me, it's ST91. In California, again, just recently, 7 years, there was a scope that scopes were improperly cleaned. ECRI, which I would address, is really like the consumer watchdog for us within the healthcare industry. And then again, on Friday the 13th, the FDA put out a warning letter and a recall. Next slide, please. So Friday the 13th, this is something that it's just unbelievable. What happened is that the FDA sent out some warning letters that there have been some issues with one of the major AER companies. And I put the link there for you to go and get the detail. But this is something that just didn't happen. If you go and you read the different links that I put here, goes all the way back that even into August they received 21 concerns or complaints about this AER, and then finally they put out and they asked people to start looking at transitioning from that type of AER to something else. Again, this is all from the FDA. Next slide, please. So, what has now happened? ECRI every year puts out their top 10. And I'm going to talk about number 1 and number 5. Next slide, please. Number 1, it's highlighting flexible endoscopes. Over the last 4 years, this has been in the news with ECRI. And I now, if you could go to the next slide, please. I'm highlighting some of the information there. And if you notice, I've highlighted some of it in red, and it says a key aspect of effective reprocessing is cleaning biological debris and other foreign material from instruments before the disinfection or sterilization step. And then if you read lower, it goes, facilities need to emphasize to their staff that inattention to the cleaning step within the reprocessing protocol can lead to deadly infections. It's about cleaning, 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 getting it clean, follow the IFU. If you follow it, you will do a good job. Next slide, please. Number 5, even though it says dealing with operating room, it's insufficient training of clinicians. Could you go to the next slide, please? This is what's very important, and I don't want you to focus on operating room. It's what I've highlighted in red that is very important. The ECRI Institute establishes that approximately 70% of accidents involving a medical device can be attributed to user error or the technique of use. This is why I've highlighted this. This gets back to how are we training people? Do they understand what they're doing? Why are they doing something? And are they following the IFU? Next slide, please. Now, Becker, along with many other publications, have published information about compliance and what's going on with it. As we know, whether it's Joint Commission or AAAAC, whoever you're using, they are coming in more and more and looking at your sterilization, your cleaning process. And if you look at this slide, notice 2011, 36%, 42% in 2012, 47% in 2013. I don't have 2014, which will be out soon because it takes time to get this. But where are they looking? They're looking at your cleaning, sterilization. What are you doing? How are you doing? Are your IFUs there? So the surveyors are focusing on this area. Next slide, please. So with this, why talk about endoscopes? I want to let you know in the bottom right-hand corner of this slide, I want to put a shout out to Corey Ofsted and her group. I'm going to use some information from much of her research that's been out there, but she has been on the forefront of research along with Dr. Michelle Alpha out of the University of Manitoba. Their information is just helping us get better and better at what we do. And what this slide is talking about, it's starting to make us look at, some people are saying there's only 1 in 1.8 million procedures that there's going to be a problem. But we have to look at this. Most outbreaks are not published. Most outbreaks are not investigated. They are difficult to link. Deaths have now been associated with poor reprocessing of flexible endoscopes. So this is in the news. This is what you're facing every day. Next slide, please. So this next slide that comes up, and I do want to apologize, it takes a couple seconds sometimes because we have so many people on here for the slide to come up. So with this slide, we have been told that it's been no big deal. There is not a risk or a low risk of infection. I believe differently. I believe we are not connecting the dots. It's not just what's found in peer-reviewed literature, it's what's also found in non-peer-reviewed literature. And that's what's important. And they don't always match. There are more outbreaks, more patients being recalled than what's being put in the peer-reviewed literature. And we know right now the focus is on ERCP scopes, but that's only the tip of the iceberg. Now, if you start reading and what's going on, bronchoscopes are starting to be put in the crosshairs. And we have to understand, it is our patient. Where is our patient getting their information? From the TV, the internet, from word of mouth. If your hospital or ambulatory care center gets into the news, does a patient want to go there? They might not be reading the peer-reviewed literature, what's out there, and to make informed decisions. They're looking at what's out in the newspaper. So let's take a quick review. Next slide, please. This here would be something from a peer-reviewed literature, and it's the damages that happens from scopes. If you look at the distal tip of that scope should not be used when you do visual inspection. It should be pulled out. And because of that, there can be infections associated with poorly cleaned scopes and poorly damaged scopes. And if you want to read this great article, there's the information there. Next slide, please. This here is just taken from all the headlines that we're being bombarded every day. Deadly bugs, somebody's passed away, superbugs here, metal scope tied to an outbreak. This has been going on, and it's not just been in this last year. Next slide, please. This here is just taken from headlines from 202 to 206. I could have made this many, many slides, but look at where it is. Patients being notified, 2,000, 1,300, 146. And look at what the reasons they found out. Inadequate, auxiliary channel not clean, failure to disinfect, wrong adapter. And it's not one area of the country. It is all over, and it has been happening, but only because of the deaths and what's been going on with the ERCP scopes has this been brought to the forefront. Next picture, next slide, please. This here is, I think, ground-shattering information. Again, this was at ISHM at their national meeting, and if you get a chance, I'd say go to ISHM next year at 2016. It's in San Antonio. It's a great place to get great information. This was on one of the poster sessions. And for those of you who don't know what the Hawthorne effect is, it's basically if somebody is watching you do something, they're going to do it right. Well, people here were actually being observed and seeing if they could process a scope properly. And even while being observed, only 1% of the scopes were actually done correctly or completely. And look at what was missed. Unbelievable. The next slide, please. So now you are a clinician out there and you're working, and there's regulations, standards, and guidelines. And what are they? I want to give a brief introduction of this, and this will segue or lead into where Mary Ann's going to pick up about ST91. Regulations are a rule or directive made and maintained by an authority. And in this case, we think of CMS. And we all know that we need approval and we need that certificate so we can get reimbursed. And CMS can come in and they can audit you. Then we have standards, recommendations that are there to ensure consistency that's fit for purpose. They are voluntary, but they can come mandatory. And in fact, CMS could look at those standards like an ST79 and ST91 and start using that in their audit process. Then we have guidelines and recommendations and technical information reports. And these are voluntary. And let's go to the next slide, and I'm going to address that a little more. So you have such as SG&A and AORN. SG&A is supposed to come out with some new guidelines, and AORN, they're coming out with some new guidelines on flexible scopes. My understanding is it should be at the end of the year or at the beginning of the year. And again, if you can go to these meetings also, it's a great way to network. But again, all of these different guidelines are coming out from these professional organizations. Then, of course, there's our CDC guidelines, which were last updated in 2008, and hopefully they'll be updated sooner with all the new information. If we could go to the next one. So when it comes to endoscopy, what are the standards that you really should be familiar with that surveyors might be using? Well, of course, there's ST79, and within that there's cleaning, disinfection. Then there's ST58 and ST41. This is 58 if you're using chemical sterilization, high-level disinfection. This is information you need to know. The surveyors know it. And if you're using ethylene oxide, that's standard 41. So these three, now you combine them and dovetail them into ST91, which is groundbreaking because now there's finally something out there to help you get to and maintain the best practice. I'm going to hand this over to my partner, Mary Ann, and she's going to try to walk you through as best we can. Mary Ann?
Good afternoon, everyone. Thanks for having me on the call today. We're going to pick up with some details on ST91, which you see a photo of the cover of the document, which just came out in April of this year. And ST91 gives guidance and best practices on flexible and semi-rigid endoscope reprocessing in healthcare facilities. What's great about this document is that it does provide reprocessing information and best practices regardless of the setting that you're in. So this does apply to any setting, whether you're in a hospital, an ASC, a physician's office. The same standard of care is applied and found throughout the document of ST91. Next slide. So within ST91, you'll find the different categories of reprocessing that we're all used to. So that includes sections such as pre-cleaning, leak testing, manual cleaning, packaging if indicated, storage of endoscopes, high-level disinfection, liquid chemical sterilization, and full sterilization practices. This document is applicable to, again, any flexible or semi-rigid endoscope, so that includes not only the GI scopes, but also things like bronchoscopes, ENT scopes, ureteroscopes, you name it. It does exclude anything that's a rigid endoscope. Next slide, please. There is a great load of information found in ST91. Things like a definition section, endoscope reprocessing design of the areas where reprocessing will occur, different considerations for personnel, cleaning, high-level disinfection, use and selection of automated endoscope reprocessors. There's a lot of information in there on questions that you as a facility should be asking when you're looking to purchase new AERs. There's also info on liquid chemical sterilization and gaseous systems such as ethylene oxide and vaporized hydrogen peroxide, information on how to reprocess different endoscope accessories, storage, transport, and a very large portion of the document is built around quality control and quality process improvement. So we've really stressed in ST91 how to build quality into your process so that you don't end up with a poor outcome. Because if we engineer quality in to begin with, then we have a much higher likelihood that we will have a good outcome in the end. Next slide, please. So these are the sections in order of reprocessing an endoscope. And as you're used to, you see pre-cleaning, leak testing, manual cleaning with the detergent solution, rinsing after cleaning to remove the detergent and any residual debris that has been loosened. And then a section that's been added in ST91 is a recommendation for monitoring the cleaning process. And we will discuss that in greater detail later on in the presentation. The next section would be disinfection or sterilization for the endoscope, rinsing after disinfection to remove any residual chemical, and then also drying an alcohol flush for the endoscope, and then also storage. Next slide, please. This is a very very high level of some recommendations that are found in ST91 that you and your facility may not be currently doing or may not be familiar with. So again, this is just high-level recommendations. I do recommend highly that you get a copy of ST91 for your facility so that you can delve into this in much greater detail. And you can do that on the AAMI website. That's aami.org. But just as a high-level review, there are recommendations that the reprocessing technicians in your facility are certified. There's also recommendations to monitor the manual cleaning process for endoscopes and also to monitor the automatic cleaning process for endoscopes. There's also recommendations to monitor water quality used in reprocessing and to monitor the temperature of chemicals that are used. This could be your detergents, could be disinfectants, anything that has a temperature recommendation on the label. There's also a new recommendation that's been found in professional society recommendations outside of the U.S. prior to now, but now we've included it in ST91 as a recommendation for the United States, that all detachable valves or buttons should be kept together with the endoscope as a unique set through reprocessing. So this would allow the valves that are used with the endoscope during the procedure to be held together as a unique identifiable set in case there is an infection outbreak, that you could pull not only the endoscopes out of service, but also any associated valves or equipment that's used with it. And that would help to identify those units very quickly. Another highlight of ST91 is a risk assessment and recommendations for when to do risk assessments, with a large one in there being around how to determine a storage interval or hang time for endoscopes. And that section of the document does walk you through how to do a risk assessment to create a storage interval for your endoscopes. And then there are also information in there on proper documentation for each category, so what you should be documenting during cleaning or during leak testing, and how to build quality into each of those different categories. Next slide, please. Now we're going to look at some best practices as mentioned in ST91. Again, this is just some of the information that's available in ST91. We'll start with pre-cleaning. Pre-cleaning is the procedure that initiates the cleaning process, and it's performed at bedside immediately after removal of the endoscope from the patient. So again, we're still in the patient room at this point. And the purpose of pre-cleaning is to reduce that level of bioburden, so whether that be patient debris or soil and microorganism that's on the endoscope, to reduce the risk of biofilm development. Pre-cleaning is required each time the endoscope is used, and it should be done immediately, again, after removal from the patient. We need to wear proper PPE, and PPE is discussed in great detail in ST91. And for pre-cleaning, we do this, again, immediately after removal from the patient, and we're going to use the cleaning solution that's recommended by the manufacturer of the scope in their instructions for use, or IFU. I saw that was one of the questions that was raised already. Some scope manufacturers recommend detergent, some recommend water, so please note that in the IFU. You're going to wipe the endoscope down with either a low linting or non-linting cloth soaked in the cleaning solution from the control section to the distal tip, and suction solution through the lumens of the endoscope, flushing all the channels, whether used or not used. Next slide, please. After we've performed pre-cleaning and utilized all of the recommended adapters that the endoscope manufacturer has provided with the endoscope, the scope is now ready to transport to reprocessing. And this could be done in many different areas in your facility. It could be in GI reprocessing, it could be in the bronch lab, could be in central sterile. So it's very dependent on how your facility is set up. But we now need to take that endoscope from the patient room to wherever reprocessing is being performed. And the recommendation in ST91 is that the contaminated endoscope, because it is highly contaminated still at this point, be transported in a closed container that's labeled appropriately, and we do have a biohazard label there, in accordance with the recommendations by OSHA and your facility. So we do want that closed, sealed container, preferably rigid, to contain the endoscope and any contamination so so that it's not exposed to the environment or any personnel. And we see an example of a transport trolley system there on the right of the slide and different bins that are available. Next slide. Following with transportation, I have some pictures here of improper transportation. On the left, it's in an unmarked container or bag, so we really have no way of looking at it and knowing, is this clean, is this dirty, are we going to return this to reprocessing, is this ready for a patient? And then on the right, we see one that's labeled biohazard, and this represents a better way to label your endoscopes in accordance with ST91 recommendations. Next slide, please. Again, we must be clearly marked for transportation, whether the endoscope is clean or dirty, and you see photos here of some systems that can be used to ensure that we know by looking at an endoscope whether it's clean and patient ready or whether it's dirty. And that is a recommendation in ST91, that you should be able to look at an endoscope and readily identify what state it is in. Is it clean or does it need to be reprocessed? So these photos are showing you transportation bins and ways of identifying. Next slide, please. So now we've transported the endoscope to the reprocessing room, and we pick up there with leak testing. This is the first step once we hit our reprocessing area, and we need to check the endoscope by leak testing every time it is used. So flexible scopes each time as recommended by the endoscope manufacturers. So we need to follow their IFU or instruction for use on how to do that properly. And leak testing is performed prior to immersing the endoscope in the detergent solution during cleaning to ensure that it's watertight and it's not damaged in any of the interior or exterior places on the scope. And there are different ways to perform leak testing. And in AAMI ST91, the different methods are outlined as a general procedure. So if you're doing a wet submersion leak test, there are instructions in ST91 on how to do that. Or if you're doing a dry automated system, there are general recommendations in there for that. So again, there are different ways to do it, but I do recommend that you make sure that the way that your facility is performing leak testing is the way that is written in the IFU of the endoscope, and you make sure you use a compatible system and it's not something that is not valid for the leak tester that you're using. The basic process for leak testing, the most common way to do it is submersion in clear water. Again, we're not in detergent at this point, we're just in water. And we submerge the endoscope that has been insufflated, so it has positive pressure, airflow going to it. And then we submerge it under the water and we look for bubbles. And it's very important that you visualize the endoscope for the period of time at least that is recommended in the endoscope instructions for use. And that normally is at least 30 seconds, but we want to look at the endoscope underwater and look for bubbles while angulating the distal tip, pressing the buttons, all to see if we find any slow leaks or large leaks. Next slide. The next slide shows a photo of the internal spaces of an endoscope, and this is why you leak test. This shows you the corrosion and buildup that can happen in the internal spaces of the endoscope if it's not leak tested. So if it is submerged and there's a leak, that fluid can flow into the internal spaces and you will have corrosion and you will have a lot more damage to the endoscope much more quickly. Next slide. So now we've done leak testing and we've passed our leak test, and we move on to manual cleaning. Manual cleaning is the most important step. It will remove over 99% of the bioburden on an endoscope, and we perform manual cleaning for every scope, every time, for every channel, whether it was used or not during the procedure. And we also need to manually clean any accessories, such as the valves that are used with the endoscope. To perform manual cleaning, you will submerge the scope under the level of the detergent solution. So again, leak testing was performed in water. Manual cleaning is performed in a properly diluted detergent solution. If we have an endoscope that has a water-resistant cap, we will have already put that on prior to leak testing. So we want to ensure that that stays in place for the whole reprocessing procedure. And to perform manual cleaning, we've submerged the endoscope, and we're going to brush all external surfaces of the endoscope with a lint-free cloth or appropriate sponge and wipe down that distal tip, and then brush Any channels that can be brushed with the proper size brush must be compatible with that endoscope size channel. We need to brush the valves too. And if we're using a single-use brush, it should, of course, only be used that single time. Or if it's a reusable brush, it needs to be disinfected or sterilized in between uses. So we can't be using the same brush all day long for a flexible endoscope. During this process, we also need to flush all channels with detergent. There is a requirement for suctioning from many endoscope manufacturers during manual cleaning. So please note that in your endoscope instruction manual, what applies to your specific model of endoscope. It is during the process of manual cleaning that you may be using an automated washing or cleaning device, such as an automated pump, and there are several different ones available on the market that will automate the cleaning process, so it will flush detergent, water, and air through the channels. So if you are using an automated system, this is where it fits into the process. Next slide. As I mentioned, you must use the right size brush, and it is absolutely necessary to ensure that you're using a properly sized brush for your endoscope channel. And there is a recommendation for the type of brush that's to be used, and there is a channel marking on endoscopes to show what size channel it is. You can't use just one brush for all of your endoscopes. You have to know that it's properly sized in order to create the friction against the walls of the lumen of the endoscope to remove that debris. So if it's too small, you're not going to have that friction at the surface of the channel, and you will not scrub the debris off of the channel. Next slide. This next slide just shows you some photos of what improper sizing of brush looks like in a lumen. So if it's too small, it won't contact or have friction against the walls of the channel. If it's too large, you'll see how it pokes out of the end of the channel, and you won't have proper friction created. But one that is properly sized will create the correct level of friction and remove the debris from the endoscope channel. Next slide, please. This slide shows you a photo of the IFU or endoscope brush guide that Healthmark has that helps you determine what brush size to use in accordance with the endoscope color guide. So there in the photo you see a yellow color, and you'll see a yellow on the chart of the brush guide to see that you are choosing the appropriate color brush for the diameter of the channel. Next slide, please. And speaking of brushes to be used, we have a close-up here of an elevator area of a duodenoscope, also called an ERCP scope. And note that this scope does need a special brush, or two brushes in most cases, to fully clean around that forceps elevator recess and around both sides of the forceps elevator. Next slide. This is a photo of the ERCP cleaning brush that is utilized for cleaning around that forceps elevator in a duodenoscope. And we do see two different style brushes that are there for cleaning around the forceps elevator. Olympus came out with new recommendations earlier this year to include this smaller brush to really clean around both sides of that forceps elevator. So you want to consult your IFU and make sure that you are using the appropriate brushes, and that smaller brush should be used for each and every cycle of reprocessing the duodenoscope forceps elevator recess there with the MAJ 1888 brush or equivalent for cleaning this area. Next slide. It's just a photo here of how to get around both sides of that forceps elevator recess in order to ensure proper brushing in your ERCP or duodenoscope. Next slide. So now that we've completed manual cleaning by brushing and flushing the forceps elevator in your duodenoscope and your normal channels in your regular endoscopes, we've now moved on to rinsing. So rinsing has to occur after the manual cleaning to remove the chemical or detergent that's been used for the cleaning process and any debris that's been loosened during brushing of manual cleaning. If the debris and the chemicals are not removed from the endoscope, it does present a risk to patients and a risk of one chemical interacting with another, because remember, we have yet to submerge the endoscope for disinfection. So this is just the detergent that we've worked with up till now. So we do need to remove the chemical and the soil that we've loosened during brushing prior to moving on to disinfection. And we do need to note on the detergent label how to properly rinse. It may say on there a specific number of rinses or the method for rinsing. So please follow your detergent manufacturer's information for rinsing and also the endoscope manufacturer on how to properly rinse after cleaning. And as far as water quality or purity goes, I can recommend that you go to AAMI TIR 34, which I see a picture of there, which is a technical information report put out by AAMI that will guide you in what quality of water to be used during each step of reprocessing. Next slide. So now that we've completed manual cleaning and rinsing, there's a new step in the process in accordance with AAMI ST 91 and 79 to verify the cleaning process. So again, this is after manual cleaning, prior to disinfection, or even prior to sterilization if you're going that route, to verify that the endoscope has been cleaned properly. We need a properly cleaned endoscope to ensure that disinfection or sterilization can occur properly. So the recommendations that are in ST 91 include an enhanced visual inspection and testing of equipment associated with it. It does have recommendations for using lighted magnification, looking at the scope using magnification for any organic residue or any signs of wear and tear such as cracks or integrity issues with the device. And if those are noted, the scope needs to be pulled out of service so that it's not reused on patients. There are additional recommendations to verify cleaning by using a method to measure either organic debris or residues that are found on scopes. And there are a variety of ways to do this. There are different swab methods available, different flush methods available, such as protein, hemoglobin, carbohydrates, detecting specific enzymes for certain types of bacteria, ATP tests, and possibly other tests in the future. And if you look in the literature, and we will discuss in more detail briefly in this presentation, there are a lot of different articles that are out there on these different types of tests. So it is upon the responsibility of the facility that you're working at to look at the different information that's available and decide what works best for your facility. Next slide. Next slide shows photos of different visual inspection tools that are available that your facility can use to really take a look at the endoscope. On the right, we have the lighted magnification that can look at the distal tip, look for any damage, look for organic debris, and really give you an enhanced visual verification that you wouldn't have had before. And on the left is a photo of a flexible inspection scope. So this is actually a borescope that can look through the openings on the endoscope, and you can see debris inside the channels. Next slide. So how do we know what markers to look for if we are using cleaning verification as recommended in ST 91? Well, if you're looking through the literature does say that the three most predominant contaminants that are found in bodily fluids or markers of patient debris are protein, hemoglobin, which is blood residue, and carbohydrates. And we know that the regulatory authorities, such as FDA, do look for results from that type of testing from the endoscope manufacturers. So they may look at a test soil such as blood residue, which is hemoglobin, or protein when these endoscopes are submitted initially to the FDA. Dr. Alpha has published these results here that we see for protein, carbohydrate, and hemoglobin, and shown that those are excellent markers for cleaning validation and verification. We do have those sources cited at the bottom of the slide. So users may want to look at testing for the same markers that are listed as what the FDA looks for, what Dr. Alpha has published, and you should understand what the test tells you when it passes and also what it's telling you when it fails. Next slide, please. All right, this slide shows you some different photos of methods that are available to assess your cleaning and to verify that cleaning has been done properly. So we have a sample from the channel, and we can test that for protein, carbohydrate, and hemoglobin, and there's one test method available on the left there that's called channel check, and test for all three of those soils together in one flush sample. Then we also have samples in the middle photo there where you can test for residual protein or blood. And then we also have ATP systems that are available that will measure any residual adenosine triphosphate in the sample that you've collected. Next slide. EndoCheck, as photographed here, is a swab test that's available, and this will look for either blood residue or protein residue inside the endoscope. And when you swab through the channel, you then will have this detection by a color change that you can see. So it's pre-packaged this way. It's an easy-to-use test with a detection limit down to 0.1 microgram for blood and 1 microgram for protein. So this is one test method that's available to your facility to check for residual blood or protein inside the channels of the endoscope. Next slide. This is more information on channel check, which is available again to check for residual organic soils, and it is often used in flexible endoscopes because it's a flush method where we flush sterile water through the endoscope and then use a dipstick test, like most facilities are used to using a dipstick test for their minimum recommended concentration of their disinfectant. This is similar to that, as you dip it in the water and look for a color change on the pad to determine if there's residual blood, protein, or carbohydrate. And I do have the information on this slide of a study that was done by Dr. Alpha at the University of Manitoba looking at using channel check on testing of scopes to verify cleaning. Next slide. The next slide is on ATP testing, and there are many different methods or different systems out there for testing ATP. There are flush methods where you can flush water through the endoscope and test that for the level of ATP, and there are different swab methods that are available for ATP that can be performed on surfaces or swabbing of lumens. It just depends on the system that you're using. There are many articles available in literature about ATP, some of them being positive and on the use of it, and some showing that it's not so positive for use in facilities. So I've noted some of those articles here for your reference. Okay, now we're going to attempt to do a poll. So you'll see this question come up on your screen here. And the question is, does your department use any type of test to routinely monitor your flexible endoscope to see if it has been properly cleaned? Again, this is not culturing. This is cleaning verification. So please, if you're able, click on the yes or no button, and we'll get a poll of the attendees to see if you're currently monitoring your cleaning process. Okay, so far we see it's about 50-50. And that's great to know. So again, as I outlined in the presentation, there is now a recommendation in AAMI ST91 that says weekly, preferably daily, you should be monitoring your cleaning process to ensure that endoscopes are being cleaned properly prior to disinfection. So thank you for answering that poll. Next slide, please. So now we've completed cleaning, we've completed our cleaning verification test. The scope moves on to either disinfection or sterilization. So we're going to look at disinfection because it is still the standard of care for flexible endoscopes. So let's look at some key points that are outlined in ST91 in regards to disinfection. We know that label claims of disinfectants can vary. So there could be different numbers of rinsing that need to be performed after soaking the endoscope. They vary in their contact times from very short periods of time up through 45 minutes. So we want to make sure that we are using our disinfectant according to its label claims. So you need to refer to the label of the disinfectant for that information. There are different types of disinfectants available, single use or multiple use. And if it is multiple use, you have to use the solution test strip to verify that you're above the minimum recommended concentration. For multiple use disinfectants, you also need to make sure that we are monitoring those label claims and not going beyond the maximum reuse life of that disinfectant, which could be as short as 5 days up through 28 or 30 days. So they can only be used till up to that maximum reuse life or until it fails test strip, whichever comes first. And that's outlined in ST91, along with the recommendation not to top off solutions. All parts of the device do need to be submerged in the disinfectant, and if you're reprocessing them manually, then the channels do need to be filled with disinfectant using a syringe in order to make sure the internal spaces of the channel are perfused with fluid, with the disinfectant fluid. And then we let it sit for the required contact time and at the proper temperature as noted on the label. And after that, then, it's recommended that you purge the endoscope with air to remove residual disinfectant, and then we move on to rinsing. If you're using an automated endoscope reprocessor, much of this is automated. So this really is for if you're manually reprocessing. But we need to make sure we're removing all of that residual disinfectant from the endoscope and the channels. And different disinfectants have different rinse requirements. So it could be that you have to rinse it 3 separate times, and that's in accordance with the label. So we want to make sure that we have the right number of rinses. There's also a recommendation in ST91 to inspect the device prior to use. So we're looking along the way of reprocessing to make sure that we don't see residual debris and that we don't see damage to the endoscope. Next slide, please. We're moving on now to storage and moisture in endoscopes. And this is really an up-and-coming area that's having a lot of emphasis placed on it lately. We've never really thought in the past much about how dry our scopes are, but I think you'll see in the future, as more and more data is available, that these endoscopes really need to be dry prior to going into storage. And if not, we can set up that foundation for biofilm to form. Next slide, please. We need to keep these endoscopes dry. So from a microbiology perspective, an absence of moisture in the endoscope or in the channel means that bacteria can't replicate, and thereby we prevent biofilm from forming in the first place. So I want to make sure that everybody understands that. These endoscopes have to be completely dry prior to going into storage, and if not, they can be recontaminated by the environment or the water, the water that was used in the endoscope after disinfection, in order that the scope is not recontaminated by the environment or that water. Next slide, please. Here we have a picture of improper storage, and there's a lot of information around storage from professional societies that show you and tell you, along with the endoscope manufacturer recommendations, that they need to be hung vertically, not coiled, and they should be not touching the bottom of the cabinet, and they need to be completely dry. And as far as testing the endoscopes in storage, traditionally the only way to test a scope to show that it has not been contaminated is through culturing. And we know that this is labor intensive, time intensive, economically intensive, so culturing can be difficult for flexible endoscopes to show that they're patient ready. Next slide. We will talk about culturing later in the presentation. Next slide, please. So we said that they need to be hung vertically with the distal tip hanging freely in a well-ventilated, clean area. They should not be touching each other. Any removable parts, such as valves and caps, need to be off of that endoscope to facilitate airflow. And the detachable parts, such as those valves, should be stored together with the endoscope. That is in the new AAMI standard, again, so that we have a unique identifiable set in case there was a problem or an infection outbreak. And there are different devices that are available to your facility to help with this recommendation of ST91, such as a small bag or a similar containment device that would keep those scope parts together with the endoscope. Next slide. Following along with storage, ST91 recommends that there be a way to readily identify if an endoscope is patient ready. So you should be able to look at that scope and see that it has been properly reprocessed, when it was reprocessed, and if it's in storage, how long it can be in storage prior to having to be reprocessed again. The photo here shows an example of some tags that are available to clearly identify that a scope has been reprocessed, that it's clean, it's disinfected, and it also captures information such as when it was cleaned, who did the cleaning process and disinfection, and also when the high-level disinfection or reprocessing occurred. Next slide. In the recommendations for storage, we said that the scope should hang vertically and not be touching other scopes. So this photo shows one type of distal tip protector that is available that will keep your scopes from touching each other. while still facilitating airflow through the channel. So there are different products that are available. This one in particular, it's not a spongy material, so it won't remain wet if there's any moisture there to begin with. So it will help facilitate the drying and allow for airflow through the channels while protecting your endoscope. Next slide. This slide shows another product that's available to keep the valves together with the endoscope as that unique set that's mentioned in ST91. And we have a picture on the bottom there of ways that we see many facilities currently storing their valves. And if there was an outbreak, there's no way to know how these valves and cleaning adapters, what was used with what particular model endoscope for what patient, because they don't have serial numbers on them. So there are different ways that you can keep those valves together with the scopes to meet those requirements in the new standards. Next slide, please. Here we see those valves and different cages that are available for keeping the valves together as it's mentioned in ST91. This particular model that's shown here also keeps the air-water channel cleaning adapter that's used during pre-cleaning with certain models of endoscopes to allow that to remain together to be uniquely identified and pulled out of service if necessary. Next slide, please. Oh, we have a poll question. I'm sorry. So currently in your facility, do you keep your valves and other accessories together as a unique set, as is recommended currently in ST91? So if you would please select yes or no, I would appreciate that. And then we'll get a poll of what facilities are currently doing. All right, great. So 68% of facilities are not keeping their valves together. So that may just be because you're not aware that this is a new requirement in ST91, or as a follow-up poll, if you selected no, please let us know with the poll if this is because you're using single-use valves. Let's see if that poll comes up. Oh, I'm sorry. Let's take a step back there. So if you are keeping your valves together as a unique set with the endoscope, can you please tell us how you're doing that? Are you using a mesh bag, a valve-safe cage type device, a peel pouch, or some other device? Okay, so by far, facilities are using a mesh bag or other. As a follow-up to this question then, we do have, if you answered no that you're not using single-use, if you're not using a containment device, are you using single-use valves? Okay. We see about 31% are using single-use valves, whereas about 69% are not. So thank you for answering those poll questions. We're almost done with my portion of the webinar. So that brings us to culturing. So this is a question I receive very often on should I culture my endoscopes? And there is guidance that's been put out earlier this year from the CDC. It is an interim guidance on how to culture specific to duodenoscopes at this point. But if you do have a question on culturing, I will refer you to that CDC method, which says that if you're culturing your duodenoscopes, you should be culturing that instrument channel, which is a suction biopsy channel, and the distal end, which is that elevator recess and mechanism. And if you have an older model elevator, an older model duodenoscope that has an open elevator channel, that you would also culture that. On the newer model duodenoscopes, that channel is sealed and cannot be flushed to sample. So as a poll question, and our final poll question, can you please tell us, are you presently culturing any of your endoscopes? So this would be on some sort of routine basis, periodic monitoring. Okay, thank you. So we see about 22% of those on the call are culturing endoscopes on some basis. And again, we can't tell with this what the basis is. And 78% are not culturing endoscopes. Next slide, please. So to finish out my portion of the webinar, thank you so much for tuning in today. I do love to be able to talk to everyone. I'm going to finish out with quality and process improvement. And this quality and engineering quality into your reprocessing procedure and continuous quality improvement is so heavily talked about in ST91. There are ways to engineer quality into every step along the way to ensure that you have the best outcomes and that you're improving your process and looking at what can be done in each step along the way as far as documentation and quality performance indicators throughout each stage of reprocessing to ensure the best outcomes for your patients. So there are also risk analysis information built into ST91 and how you can do a risk analysis around things like interval for storage of endoscopes, also known as hang time. So it's so important to me to stress to you to build quality into your process up front before there is an issue to ensure that we have the best outcome for our patients. So thank you so much for having me today, and I'm going to turn it over to Steve, who's going to finish out the webinar. Thank you.
Thank you, Mary Ann. To everybody out there, I don't know if the facilitator, there was one poll question that didn't get brought up, and if it's possible, can you pull it up? It was dealing with, yes, thank you. If you answered yes, what type of test are you using to check for the cleaning verification process? If you could click on that, please, and we'll finish up the presentation. Thank you. I'm sorry it's out of order, but it didn't come up. If you could answer that for us, please.
So if you are using cleaning verification, what type of product are you currently using?
We'll give you a couple, about 30 seconds to fill that out.
Okay, so we have the majority of callers using channel check and then ATP and other. Okay, thank you for answering that.
Okay. Now, I want to close with, and I put no silver bullet, and the reason I put this in here is that an ounce of prevention can yield enormous return for the patient. I want people to understand that implementation of any quality improvement or risk-based program does not always prevent incidents from happening, but it will help you reduce and understand those incidents better if they do occur. We've presented a lot of different tests. It takes a lot of different tools in your toolbox to put that picture together if you're doing a good job in getting to best practice. So could you go to the next slide, please? Again, we hope that this program will give you some ideas and thoughts to get to and maintain best practice. So if you go to the last slide, please. We've only given you the highlights of ST91. It is over 200 and some pages. If you haven't bought 91, go to www.amy.org. Also, remember, a policy should be based on the various standards and recommendation for flexible scopes along with data from scientific data. Remember, users verify, manufacturers validate the process. We cannot stress the importance of training. Your staff needs to be certified. Take the certification test, wall charts, whatever you need to do competency. Each facility has to adapt a policy to their own situation. So if you go to the last slide here for questions, I know we only got about 2 or 3 minutes left. We flagged a couple. They're going to add quickly as we can here.
Sure. Hi again, everyone. Thank you, Steve and Mary Ann, for that informative and enjoyable presentation. Unfortunately, we only have time for about one question, but our presenters can follow up on questions they don't have the opportunity to address because a lot have come in from our audience. So we can start or finish also with this one. What hang time or how many days are you comfortable with to reprocess scopes before patient use?
Mary Ann, go ahead.
Okay. So this is a tough question that we get all the time, and as part of the AMY committee recommendations, we really discussed this in great detail and went back and forth, and we ended up not coming up with a specific recommendation because it's so dependent on how the endoscope is reprocessed, what the verification process is for cleaning around that endoscope, and then how is it stored? Is it stored in a cabinet? Is it properly dried? Are the scopes retrieved with gloved hands? So there are so many variables to account for that. What the AAMI committee decided to do was to put out a recommendation to do a risk assessment. So you have to look at the procedures in your facility to know what works best with your set of variables to have the best patient outcome. So I don't have a number of days associated with that, but really looking at your specific policies and procedures and variables in your facility to ensure that you're coming up with the best set of recommendations as far as how many days a scope can hang before it is reprocessed. And I just want to reiterate that we do have a list of these questions, and Steve and I will work on those, and we'll have your contact information and get back to you on the other questions that you've asked. And I apologize that we don't have time to take all of those. We did get a whole slew of questions in the chat box.
One quick question. IFU stands for Instructions for Use. How about we close on that, guys?
Great, perfect. Thank you, Steve and Mary Ann, for your presentation today and for all of our audience members for participating. We look forward to having you join us for future webinars. This concludes today's program. Have a wonderful afternoon.
Bye.
Bye.